Continuing on with epithelial tissue, we'll start looking at the individual types of epithelial tissue. First up is simple squamous epithelium. Now, in the case of simple squamous, we're looking at simple, one layer, squamous, flat cells. So as you can see here, the diagram shows a layer of simple squamous. The histology slide, you look at each individual one is creating a layer. This is one layer of simple squamous. It's forming the shape around the alveoli of the lung. So each one is an individual cell. So here's one cell, then here's a second cell, and a third cell, and a fourth cell. You can see the individual nuclei in each one of those. So location, you can pick up air sacs of the lungs if you want. You can look at it as the lining inside of the kidneys. This, right here, tracing around, that is going to be the simple squamous epithelium around a kidney particle. So that's going to form basically the covering around the glomerulars, around the kidney structure there. So simple squamous, functionality, what's it doing? Allowing materials to pass through. Looking at diffusion, looking at filtration. So single flat layer allows for materials to pass through. Simple squamous epithelium. Examples great are going to be in the lungs, in the kidneys. You can even find your blood vessels, lining the inside of the blood vessels, keeping it smooth to allow the blood to flow through. There are some specialized names for certain locations, though, for simple squamous. The term endothelium, endo, inside, endothelium is going to be a friction-reducing lining. The whole purpose here is to try and make something slide past as smoothly as possible. It's the idea of endothelium. Endocardium. Well, cardia refers to heart. So endocardium is lining the heart. It's the inside lining where the blood touches the actual main chambers. So looking at endocardium. And last one at the bottom is the mesothelium. Serous membranes, that's key, serous membranes. We'll get back to those more. But these serous membranes are lining the body cavity and covering various organs. So looking at serous membranes. Simple squamous, flat, one layer, covering or lining various structures in the body. Simple cuboidal. When trying to identify simple cuboidal, it's one layer of cube or box-like cells. This is what we're looking at for a simple cuboidal, forming a ring shape. Another one down here. Each one of these rings is formed by multiple simple cuboidal cells. This is one single cell right there, a second cell next to it, a third cell, a fourth cell, a fifth cell. But they're all forming that ring-like structure that is going to be your simple cuboidal here. Now just keep in mind, apical and basal also. This is the apical surface. It's the inside. It's exposed to the inside cavity. Well, that's apical. This is basal out here. The basal is where it touches the underlying connective tissue. So basal. This particular example looking at here are going to be ducts in the kidneys. So a great example is in the kidneys. You can also find them in other small glands. For example, you find them in the thyroid. But this single layer of cube-like cells, so cube-like, secretion and absorption is the main thing here. So you allow for diffusion, for example, but also absorbing things through. So looking at simple cuboidal epithelium. Simple columnar epithelium. One layer, tall, column-like cells. Simple columnar. Now this one, there's two different examples of, because one is going to be ciliated, one is non-ciliated. So in this one over here we're looking, if you notice, there's really nothing sitting there. that is going to be a non-ciliated variation of it. But if we're looking over here, you can see the tiny little cilia are protruding from the cell surface. That is a ciliated version. So depending on where you find these, if you find the simple columnar in the digestive tract, they're non-ciliated. But if you find the simple columnar in the respiratory tract, they would be ciliated. So it depends on location whether they have cilia or don't have cilia. But regardless, they're still going to be simple columnar. Same name, just are they ciliated simple columnar or are they non-ciliated simple columnar? As far as the actions, mostly absorption. In your intestines, that's all absorption based. In your respiratory tract, it's still mostly absorption based there as well. So looking at absorption going through the tissues. Again, single layer, it's easy to pass through compared to a multiple layer. So simple columnar. Absorption, we have the non-ciliated found in your GI tract, intestines. We have the ciliated version found in the respiratory tracts. Simple columnar. Pseudostratified columnar. Now in the case of pseudostratified columnar, both these examples are going to be ciliated. We're just trying to show the different variation here. This is going to be the area that shows the pseudostratified columnar, as well as this area. Both of them have the apical, this is apical, and this is apical. And then both have the basal, basal here, basal there. So just looking at just different orientations and different locations. Sometimes it looks like you have tons of layers because you have nuclei spotted in all different places. Other times, you're going to have it look like, well, there's a nuclei here, nuclei there. It looks like maybe only 2 layers, maybe 3 layers. So it's not quite as many layers appear to be there. But these are going to be both ciliated, pseudostratified columnar epithelium. If you notice, they contain goblet cells. That's a goblet cell, that's a goblet cell. So you still have goblet cells located in these. There's another one in each one. So that means the full name really is ciliated pseudostratified columnar epithelium with goblet cells. So it's quite a long name, but when you're identifying the tissue, we're looking at pseudostratified columnar. The ciliated and the goblet help you identify what's inside of it as well. So that is your pseudostratified columnar. Location, trachea is probably the most obvious one, running right down the middle, trachea. So you have also a little bit of the upper bronchi. Basically, the trachea splits the bronchi. Functionality here. Well, if you have the cilia with goblet cells, that means you're producing mucin. The cilia can move that mucin along. So a propulsion of the mucin along the cell surface. So you can see it secretes mucus, and that moves mucus. So we're producing it and then moving it right along. Pseudostratified columnar. Here we have stratified squamous. In the case of stratified squamous, we have 2 different types. We have keratinized, and then we have non-keratinized. In the case of keratinized, keratinized can be identified because of this dry layer above it. If you notice, non-keratinized doesn't have it there. The keratinized is going to be having this entire layer that's all dry, dead, scaly cells on the surface. That's your skin. Keratinized stratified squamous is your skin. You have all these dead layers of protection on this dry covering. Now the non-keratinized, that is more of a moist membrane inside the oral cavity, lining the vaginal canal, lining the anal canal. It's where still friction can occur, but it's having a protective basis by having the multiple layers of stratified squamous. So really the biggest thing is here is protection. Multiple layers are providing protection for the underlying tissue. You still have apical and basal, This is apical up top. This is apical. The basal surface then is a lower one. This would be basal here, kind of tracing that. And this is basal. So still apical and basal, just more layers in between, a thicker layer of epithelial tissue. Moving on from this stratified squamous, we can move into stratified cuboidal. Now, stratified cuboidal isn't nearly as common in the human body, but you can find this double layer in this case. So 2 layers. We have 1 layer here, 2nd layer here. So we can find this double layer of box-like, cube-like cells can be located around salivary glands and sweat glands primarily. The idea here, because it's 2 or more layers, is more protective. So the idea of the stratified cuboidal epithelium. Now, stratified columnar, I don't have a picture of the histology up here because you really don't see it very much in the human body. But the only places you'll find this stratified columnar, so again, 2 layers or more of tall column cells. But the only place you'll find it is in the male and a little bit of the female urethra. And again, in this case, a little bit of secretion and protection as well. You'll notice that anytime it's a stratified tissue, stratified squamous, stratified cuboidal, stratified columnar, it has protective aspects to it. And moving on to transitional. Remember, transitional doesn't have a set cell shape. Instead, you have all 3 possible ones here. You can have the squamous-like cells, you can have the cuboidal-like cells, you can have the columnar-like cells. This particular transitional, looking at this, the tissue right here, is going to have all these present. You can notice that some cells are going to be very tall, like this one. Other cells might be a little more box-like or cube-like. This one's kind of cube-like. Or you get some very flat ones that are kind of squished, which as we start looking around down over here, you can see there's more of a flat one right there. So you're looking at different cell shapes in transitional, kind of all meld or build together. Still have the basal. This is the basal edge right there. And then you also have the apical, the part that's exposed or open. So this is your transitional tissue found in the urinary bladder, found also in the ureters. Ureters go from the urinary bladder up to the kidneys, or kidneys down to the urinary bladder, I guess. So what we're looking at here is the ability to stretch or expand, term distension. So stretching or distension is going to be the transitional epithelium. So those are the different types of epithelial tissues, from simple squamous, simple cuboidal, simple columnar, up to pseudostratified columnar, up to stratified squamous, stratified cuboidal, stratified columnar, and then transitional to wrap it up. All of them have those same traits we talked about in the previous videos.